Hello everyone, my name is Verónica Baronetto from Córdoba, Argentina. We are working in tumor immunotherapy with Guadalupe Nibeyro and Juan Vazquez. Our director is Dr. Elmer Fernández. In this research, we analyzed whether the use of streaming preprocessing in RNA-seq bases is necessary or not for TCR clonotype identification. The T-cell receptor TCR are responsible for the identification of tumor neoantigen present on the surface of the tumor cell. These processes are favored by checkpoint immunotherapy, leading to an effective anti-tumor response. The diversity of TCR clonotype of the tumor microenvironment can be characterized by means of bulk tissue RNA-seq data. The TCR are generated through the process of BDJ recombination at the TCR beta loci, through complex rearrangement by means of splicing, single nucleotide polymorphism, insertion, and deletion events that require specifically designed alignment algorithm for sample mixer for the identification of the sequence transcript data. The TCR identification starts with sequence raw data, FASTQ file, that should be aligning against a TCR loci reference sequence. Since sequences RIA do not always have correct signal, a process known as streaming, which removes adapter and low-quality bases is often assumed as necessary. The continuous improvements in technology, chemistry, and algorithms have led to suggest that the streaming process could be avoided. This could consequently optimize processing time and computational cost. Here, the impact of the adapter and low-quality real removal streaming is evaluated by comparing the TCR clone identification by using streaming and raw RNA-seq data. First, RNA-seq data of 10 subjects from the European Nucleotide Archive, which were randomly chosen. This dataset contains 2 cohorts of patients who were treated with different immunotherapies. Then, the data were streamed by Tringalore. Raw data was also used. FASTQ files obtained from both processes were used to feed the mixer software to identify the TCR clonotype for each sample. The identified TCR clonotype population from both approaches was compared by using the Jensen-Shannon divergence score. The overlap and count difference between the clonotype and correlation between clonotype across the study sample population. The result identified 818 and 825 TCR clonotypes in the Joule test cohort for treatment and raw data respectively, where 800 of them were shared between both approaches. The count difference in the clonotype overlap was only in 9 of these. The Jensen-Shannon divergence score ranged between 0 to 0.004, and the correlation score between paired samples ranged between 0.98 and 1. In conclusion, these findings indicate that there are no differences in using streaming data over raw data, with the consequent gain in processing time and computational cost of the latter approach. Thank you very much for your attention.